My name is Zach Hines. I'm an associate professor at CBMR, as was mentioned. And it's my great privilege to sort of close out this morning session with an introduction of Professor Satchidananda Panda from the Salk Institute in La Jolla, California. So Satchin needs very little introduction, but I think suffice to say he's been a pioneer for many, many years in linking sort of the circadian clock to metabolic health from various organ perspectives. I think for me, one of the more intriguing studies that he's done is actually look at the circadian transcription in different organs in monkeys, actually, and trying to link this between, we often study nocturnal mice, and we, of course, are not nocturnal humans, and so trying to link mammalian metabolism in a circadian manner. But perhaps most famously, Satchin's really known for establishing that you are not what you eat, but really when you eat. And so really looking at eating at certain times really confers enormous metabolic benefits throughout sort of a whole organ and whole body system. And so I think we'll probably hear some updates on the benefits of that from Satchin. So please take it away.
Thank you. And it's very embarrassing when you send the wrong slides. Okay, before I start, I would like to thank the real heroes who actually carried out most of these studies. Almost 10 years ago to this month, we published the first time-restricted feeding study that was led by Christopher Ballmers, Megumi, Luciano, and Amir, and all four of them have their independent labs now. And then Amandine followed up some of the studies, and Amandine started her lab during the pandemic, and currently Shaunok and Terry are continuing most of these time-restricted feeding studies. Although we have fly and human TRF studies, I'll not talk about that. And I'd also like to, thank the funders who are listed over there. Most importantly, Salk Innovation Grant, because most of the studies, initial studies, were actually not funded by any federal agencies for a very long time. And Hemsley Foundation, Glenn Foundation, and my disclosures are there. Okay. There's a good opportunity to really advertise for open positions, and please do apply. Salk is an awesome place, and we are also part of a large consortium called USHI, Human Performance Alliance, consisting of 6 institutes, $220 million, 10 years. So there are a lot of positions at UCSD Salk in our labs, and those positions are listed here. Okay, so almost 10 years ago, we published a paper, and the idea was very simple. What is the impact of timing of food? Because by that time, there are 9,000 papers describing diet-induced obesity, where mice or rats are given high-fat, high-sucrose diet to eat ad libitum. And around 2007, 2008, Joe Bass's lab actually published a very beautiful paper showing that when you feed mice high-fat diet ad libitum, then the mice, instead of eating 15 to 20% of food during daytime, they slightly increase their food intake during daytime by another 10 to 15%. And our question was whether this increased food intake during the daytime for mice was causing some of the metabolic diseases that are found in diet-induced obesity model. And now, if you just search, there are 11,000 plus diet-induced obesity papers. So the experiment was simple: to take male C57B6 mice around 12 to 16 weeks old, give them ad libitum access to high-fat diet. The first experiment was 63% calories from fat ad libitum, or the second group got 8 hours access to food, and they usually catch up. They eat the isocaloric diet within 8 hours. The reason why it was 8 hours was not empirical. It was because Christopher's girlfriend actually did not let him be in the lab for more than 9 hours. But anyway, so that 8 hours diet has become very popular. And what we found was, after 18 weeks, these mice which were time-restricted fed, they weighed 28% less, and most of the reduction in weight was due to reduction in fat mass by as much as 70%. And if we looked at the liver, the ad-lib-fed mice had fatty liver disease, and then the time-restricted fed mice had healthy liver. We actually sent the papers first to Nature, and then they reviewed and rejected because they said there is nothing novel here. Mice are supposed to eat at night time, and if you feed them at night time, they should be healthy. And we sent it to Science, and Science said, well, there must be something wrong because it breaks the third law of thermodynamics. And I said, okay, so which metabolism paper has actually tested the third law of thermodynamics? And anyway, so it was rejected, and I was really depressed. And then I sent the paper to Nikla at Cell Metabolism, the same Science format. I said, if you send it for review, if you promise to send it for review, I'll reformat it to Cell Metabolism. And then she said, yes, she would. But anyways, the paper actually had normal chow ad libitum, normal chow time restricted, high fat ad libitum, high fat time restricted, 4 different groups. It had the same number of calories per mouse per day. And they had very different outcomes. This paper has nearly 187 supplemental and main figure panels. And one of the key figure from here that relates to Karen's presentation just before this was time-restricted feeding reduced hepatic steatosis significantly. And this was very exciting for us because, as we know, right now in the US, 1 in 3 adults will likely get non-alcoholic fatty liver disease. And there is no approved drug so far that I know for this NAFLD. And in fact, in this first paper, we found that time-restricted feeding could prevent hypercholesterolemia, high blood glucose, body fat, body weight, improved energy expenditure, improved endurance, and motor control, motor coordination. And then Amandine joined the lab, and then we asked whether it's therapeutic or not. And she took 560 plus mice and put them in different diet, high fat, high sucrose diet, high fructose diet, and to test whether 8 hours was a magic number. So she also tried 9 hours, 12 hours, and 15 hours of time-restricted feeding. And then she asked whether the mice could ad libitum eat in the weekends so that they could, so this would be 5 days of time-restricted feeding and 2 days of ad lib. And to do the therapeutic effect, she also tested, it's a crossover trial where the mice were fed ad libitum for 26 weeks or 13 weeks, and then they're transferred to time-restricted feeding. So the food consumption was equivalent among different groups of mice. Those are compared. And actually, the 5 days time-restricted, 2 days ad lib-fed mice, those mice overeat during the weekend. So their food consumption is actually much higher than the ad lib-fed mice. But just like we had seen with the 63% high-fat diet, we also found that the time-restricted feeding of 9 hours or 12 hours was protective to some extent for weight gain. 15 hours was not that much. And this was the most important experiment where we fattened up the mice for up to 26 weeks, and then we put them on time-restricted feeding. They reduced body weight. But time-restricted feeding did not have any legacy effect because if you time-restricted feed for 26 weeks and then put them on ad lib, they quickly gain weight. A normal chow, there is usually no change in body weight. But if you look at muscle mass, then time-restricted feeding had a significant impact in increasing muscle mass in normal chow-fed mice. All those mouse experiments were done in male mice, and during that time, many clinical trials started. So we asked whether time-restricted feeding has different effect on male and female and young versus old, because most of the previous studies were also done on male young mice. So again, Amandine, during this, just before the pandemic, she did this very beautiful study where she took 2 groups of mice, young versus old, middle age actually, they're not that old, 3 months or 12 months old, male and female. And now we started feeding 45% fat, 17% sucrose, this diet, which is, people think that this is more equivalent to the Western type diet. And again, these mice catch up in their caloric intake. If we feed them for 8 to 9 hours, that's typically we do. And both male and female mice on our ad libitum diet actually eat 30 to around 30% of food during daytime. And that's what we are preventing. We are preventing reducing that daytime food intake by around 30%. And as the mice get older, they actually increase their daytime food intake. And it's very similar between male and female. They eat somewhere between 35 to 40% food during daytime. So what we found was the male mice, the body weight protection that we found actually happens to both male mice at young and old age. But the female mice don't respond to time-restricted feeding with respect to body weight. And that's kind of a damper for many people, for half of the population. But still, we said, okay, so we'll continue to see what are the impact on body composition. And again, the body composition, again, for female mice, we don't see much effect of time-restricted feeding in reducing fat mass, either in young or the old mice, but the male mice respond pretty well. And if we ask the serum leptin level, which roughly follows body fat, again, in male mice, we see significant reduction in leptin, but not in the female mice. But the scale is also very different. The female mice, as you expect, have slightly more serum leptin level. But what was interesting was, even though there was no impact of time-restricted feeding on body mass and body composition, there's actually a significant impact on liver fat. And this is just the H&E section showing both male and female mice under our liver feeding condition have some lipid droplets in the liver. And this actually exacerbates much more in older mice, and time-restricted feeding could protect that. And that's the quantification liver triglyceride level in both male and female mice. Liver triglyceride levels almost come back to the same levels under time-restricted feeding. So this is very important because we know even in humans, females are actually relatively more protected to fatty liver disease. But once they get fatty liver disease, then the progression of disease or the severity of disease is much faster. The progression is much faster than males. And we are pleased to see that time-restricted feeding has similar effect on both male and female, irrespective of weight change or body composition change. And again, irrespective of body composition change or weight change, the female mice, both young and old mice, are also more sensitive, also show improvement in glucose tolerance. Of course, the males have slightly better improvement than the insulin levels. The fasting insulin levels also go down. But as you may know, the male mice on high-fat diet actually produce a huge amount of insulin compared to female mice. So the delta is much higher for male mice, and that may be driving the significance. Then it was towards the end of the study, we wanted to see whether these mice respond differently to LPS challenge. And this is a completely out of the blue experiment that Amandine thought we should try. So we put the ad-lib and TRF mice, 12 months old mice, which have already gone through 3 months of time-restricted feeding, so around 15 months, on LPS challenge. And what was interesting was nearly half of the mice, nearly 80% of male mice succumbed to an LPS challenge within 10 days. So within 10 days, nearly 80% die, and time-restricted feeding protected nearly 50% of mice from death. Whereas female mice, the time-restricted fed mice were completely protected from LPS challenge. And that was a very big surprise for us because, again, these mice had higher fat mass. And they didn't change any body weight, but there are many benefits that were independent of body weight or body composition. So that's the overall summary, and it was published a couple of years ago. So with that, over last 10 years, we and others now have reproduced this data, and then essentially now it boils down to this theme that just like caloric restriction, reducing calories, improving nutrition quality, optimizing time can also have beneficial impact on health. But during the intervening years, we have also seen there are multiple other time-restricted feeding papers showing improvement in cardiac function, improvement in blood pressure, even cognitive tests, and the impact seems to be pleiotropic on multiple organs. So then we asked a very simple question: what are the impact of time-restricted feeding in multiple organs as we used RNA sequencing as the first pass? It's a very simple experiment. Again, this is done mostly in male, so this was exclusively done in male mice because that's where we had seen the maximum benefit. So 12 weeks of B6 mice given this 45% high fat, high sucrose diet for nearly 7 weeks, because we thought if we continue our liver TRF for a long period of time, then we'd also see the impact of the pathology. And after 7 weeks, we harvested this 22 different brain regions and peripheral organs in every 2 hours over 24 hours. And did the RNA sequencing. Out of those nearly 1,000 plus samples, 1,035 samples passed the QC. And that's the summary I'll present. So since it's a very complex data set, multiple tissues, time series, then we divided the approach, the analysis approach, to 2 different approaches. One is time-independent analysis. Just consider all those 12 time points within 24 hours are just replicates. So in that way, for every organ, you have 24 replicates because 2 mice per time point. Or time-dependent analysis, you can do time series analysis on the data set and then ask what are the temporal changes. So from the time-independent analysis, you can also get differential expression. And then very simple thing that we can ask is, what are the common differentially expressed genes in 5 or more different organs? Why 5? Because we just picked 5. There are many brain regions. There are many heterogeneous peripheral organs. So we thought 4 or 5 tissues will be pretty good to begin with. So the first thing, on a broad scale, nearly 70% of genes, that includes both protein coding and non-coding genes, or 80% of protein coding genes are differentially expressed in at least one tissue under time-restricted feeding condition. And I must also say there are many behavioral and pharmacological interventions to change metabolic health, but never before people have looked at so many different tissues. So it's possible that many other interventions also have very similar, very pleiotropic, very large effect. And so these are the differentially expressed genes in different tissues. As you can see, the biggest effect is actually in the white adipose tissues, whether it's brown fatty, what or I what. And then the second biggest effect happens in the digestive system and then in the hypothalamus and adrenals. So that means neuroendocrine cells are also affected by time-restricted feeding, at least in terms of differential gene expression. Then if we ask simply, what are the number of genes that are upregulated in X number of tissues, so 1, 2, 3, 4, you can go up to 13 different tissues, then these are some of the top genes that are differentially expressed upregulated under time-restricted feeding. And similarly, those are the ones that are downregulated. So for some reason, we actually find leptin mRNA in multiple tissues, and it's always downregulated in time-restricted feeding mice. And if we do pathway analysis, then what are the pathways that are changing in 5 or more tissues? Then it's mostly RNA and protein processing, fatty acid metabolism, ribosomes, and lysosomes. So those are upregulated in TRF, and then the downregulated pathways are here, including this BCAA degradation, which was a little surprising because we thought, well, BCAA degradation, if it is downregulated in TRF, then how come branch and amino acids levels actually go down in time-restricted feeding? Because that's what we had seen before. So we went back and asked what happens to branch and amino acids. Most of the branch and amino acids actually end up in the adipose tissues in healthy mice, whereas in diet-induced obesity model, branch and amino acids accumulate in many peripheral organs. And if we look at the gene expression, actually what happens is under time-restricted feeding, all these genes that are involved in branch and amino acid degradation, they go up only in EWAT, whereas the other ones in other organs, their expression levels go down. So this might explain that they're actually in time-restricted feeding, branch and amino acids are ending up in the right tissue and they're also degraded there. Then I'll talk a little bit about time-dependent analysis. So if we look at cycling or 24 hours rhythm and gene expression, then what do we see? So what we found is nearly 65% of genes are rhythmic in TRF in at least one tissue, whereas under ad-lib condition, only 40% of genes are rhythmic. And again, here we do see some tissue-specific effect. Some of the tissues actually respond much better to time-restricted feeding in terms of rhythmic transcripts, not differential expression and vice versa. One thing that we always thought, and that's always written in many reviews, is time-restricted feeding might improve cyclicity of many circadian transcripts, but that has never been formally tested, at least in vertebrates, like in mice. We had done some experiments in flies, in fly heart, fly head, and body, and there we had seen some evidence that time-restricted feeding might be improving cyclicity and synchronicity of rhythms. So if we take the peak time, the time of peak expression of all the cycling transcripts in different organs, and then stack them up one above another, then this is how we see under ad libitum feeding conditions, there is not a specific time when circadian transcripts actually increase they're synchronized or they're more prevalent. But whereas under time-restricted feeding, what we find is there's a clear separation, there's a clear peak at the end of the fasting. So that's around this time, and also end of feeding. Because the usual prediction from caloric restriction field is time-restricted feeding imposes a long fasting period. So you would see mostly fasting-induced genes rising. But at the same time, what is interesting is these genes that rise just during fasting, because of the circadian effect, they also begin to go down before feeding begins. And then the second take-home message is, even in response to feeding, we see another cluster of genes that cycle and reach their peak expression level. So it's not only fasting response, there is also a very strong feeding response under time-restricted feeding. And then if we ask very specifically, what are the genes, what are the pathways that are enriched here and during fasting or during feeding, we also see slightly different clusters. During fasting, most of the organs, so increase in rhythmic expression of the peak expression levels of pathways in autophagy, DNA repair and apoptosis, cell cycle regulation, and fatty acid oxidation. Whereas in feeding phase, it's mostly proteostasis and RNA processing, most of the RNA and protein quality control that happens in response to feeding. Then we asked, well, what happens to clock genes? Actually, the core clock genes, they don't change too much in response to time-restricted feeding, at least in this 40% diet. Whereas in 63% fat diet, we had seen improvement in amplitude of some of the clock genes. But here, they are overall similar at RNA level. We don't know what happens at the protein level. But if we ask, these are the major pathways that improve the rhythmicity under time-restricted feeding, and those are mostly under TRF, so those are shown here. So at the same time, we also did metabolomics from the liver samples. Again, the same 12 time points under our liver and TRF. And this is one example that what we found is TRF reduces diacylglycerol levels in liver. And these are the different species of diacylglycerol, and they're almost uniformly reduced. Most of the reduction happens because the diacylglycerol levels don't go up in response to feeding, because feeding, our liver-fed mice usually still eat 70% of their diet during nighttime. That's where all the DAG upregulation happens. And we know that DAG is linked to insulin resistance, so you would suspect that the time-restricted feeding mice should have low insulin. At least the fasting and fed insulin should be slightly low, and that's exactly what we find here and also in previous papers. Then we also looked at clusters of genes that are cycling in different tissues. And one common theme that came out was fatty acid uptake. The genes that are involved in fatty acid uptake in jejunum and ileum, those become more rhythmic. And then the genes that are involved in uptake and recycling of VLDL in liver and EU wards, they also became more rhythmic. So that's what is mostly shown here. And even in metabolomics, plasma metabolomics, we find similar signature that partly explains some of these changes. Then fatty acid oxidation also, what we see is time restricted feeding, most likely it synchronizes many of these genes that are involved in fatty acid oxidation. So they're actually expressed around the same time. And so this coherence of gene expression across different time points might explain increase in fatty acid oxidation under time restricted feeding, at least in male mice. And then similarly, the autophagy genes, many pathways, what we find is they're better synchronized, or their sequential activation might explain some of this improvement in what we find in time restricted feeding in multiple organs. So in the interest of time, I'll stop there, and I would like to take some questions. So I didn't talk about human studies, and many of you might have questions about human studies. Feel free to ask, and I'll try to address them. Thank you.
Excellent. Thank you very much, Satchin. While we're waiting for people to sort of gather their thoughts, I had a question. Actually, it sort of ties in the beginning and end of your talk. So the data you just showed on fatty acid oxidation. So in the very beginning, I think from some of Amandine's work, you showed that energy expenditure is actually increased.
Yeah, we have to, during the feeding phase.
Okay, I was going to say, so you, like in metabolic cages, you know when that happens.
Yeah.
And so I'm wondering now if you can take this transcriptomic data and, of course, gene protein, but can you gain insight into, again, what is driving that increased energy expenditure?
Yeah, so what we are actually doing is we're trying to look at mitochondrial respiration at different time in different organs, and that will give us a much better idea. Because from RNA going to explain VO2 max will be
What would you
a lot of hand
have the
waving.
metabolomics?
Yeah,
That's
yeah,
also,
metabolomics, yeah.
And then I would be remiss before asking, are these all done at room temperature, or have you also tried thermoneutrality, or what?
No, it's at room temperature. We haven't tried thermoneutrality.
One more layer, 1,000 more samples.
Well, the thing is, people always ask that thermoneutrality means how many of us, actually, even humans, we don't stay at thermoneutrality. We are at 22 degrees centigrade. So
It's also a mouse versus human, so I get the point. I believe we have a question from online.
Yeah, we have a question from YouTube from Fadestini. It goes, do you see the opposite direction of effects? So the same gene upregulated in one tissue but downregulated in another. So if so, what is the explanation for that and the consequences for drug targeting?
yeah, so we actually find quite a few genes that behave that way. They go off in one and go down in another. I think some of the genes in branch and amino acids are examples. In many tissues, they go down, and in EWAT, they actually go up. Yeah, so in terms of drug targets, I can just wave my hand because we have to go deep into it to see. Actually, that's a slightly interesting question from a different point of view, because when we see there is robust cycling of many genes, almost 65% genes now cycle under time-restricted feeding. So that means nearly 60% of FDA-approved drug targets are also now cycling. So now the question is whether time-restricted feeding will change the optimum timing of drug doses, or whether we can reduce doses of some of the. So I think that will be much more interesting to see how, particularly in many of the human trials where there are many metabolic syndrome patients who are also on medications, whether time-restricted feeding is going to change that medication requirement. So that will be very exciting.
That's very interesting.
That was really a great presentation. I have two questions. So one is, can you speculate on what is the synchronizer of some of the changes of these genes? Is it hormonal or?
There can be multiple, because one thing is under time restricted feeding, actually the body temperature of mice robustly cycle, whereas under ad lib, they're almost constant. When I say robustly cycle, the cycling is around 1 to 1.5 degree, and Uli Sibler's lab almost 20 years ago had shown that that 1 degree change in core body temperature can be a strong synchronizer. And then coming to your other questions, yes, we do see in the hypothalamus, there are a lot of cycling transcripts, and that might translate to robust cycling of some of the HP axis or HPG axis. So that can be another synchronizer.
Right, so you opened the door on this one, and I'm curious about what you think about the translation of the data from rodents to humans.
Yeah.
So could you maybe talk a little bit about that from your perspective?
Well, in rodents, most of the time restricted feeding, we control for calories to make sure that this is time restriction effect. Whereas, as you have seen in most of the human studies, particularly when you go from people who are eating 14 to 16 hours and you try to restrict them to 8 hours or 10 hours, there will be inadvertent decrease in caloric intake. So the outcome can be connected to both caloric reduction and also time restriction. So now, many of the studies actually show reduction in body weight that's connected to, that's usually as, used as a cause for observed outcomes. But then if we do regression model on some of these outcomes, then what we're finding is the improvement in blood pressure particularly is disproportionately higher than the observed body weight change, because blood pressure is an area where there is huge amount of data available, and there is nice model showing if you lose X amount of weight, what should be your diastolic or systolic blood pressure reduction. And in time restricted feeding, in humans, what we're finding is there is both systolic and diastolic blood pressure reduction. And that's very exciting. So now coming to humans, the pleiotropic effect is so pleiotropic that we think that some of the effects may be independent of body weight. So hopefully there will be much better controlled study. And Courtney Peterson has published some of the nice, very well-controlled time restriction and caloric weight stable individuals, and still there are improvement in beta cell function and blood pressure regulation. So hopefully they will pan out.
Excellent.
I think that was a lovely talk. So my question kind of follows on from that. Do you know if there's a metabolic memory of time-restricted feeding? When you show in mice, obviously, as soon as you stop it, they kind of gain it back. But are they still metabolically more healthy, and how long for, if you see, if we're going to be translating it to humans?
Yeah, so that's a nice question. Actually, we're trying to do some methylome analysis to see whether there is an imprint. In humans, it's slightly different. There is a behavioral feedback that kicks in. So usually what we find in our patients who are going through time-restricted feeding, if they self-select a 10-hour window that ends around, say, 8 p.m. at night, and then one night they stay awake and eat very late into the night, then the next day they describe what they call food hangover. So they cannot, the digestive system cannot digest the food, because we think that there is the circadian rhythm in peristalsis and digestion is so strong that they cannot digest. And then next day morning, when they have the food hangover and they feel crappy, then they know they're connected, and then they come back to time restricted feeding. So that's completely independent of epigenetic or any kind of memory. It's a very quick behavioral feedback that we see. But it'll be interesting to also see, in one study, we're actually collecting muscle biopsy from metabolic syndrome patients who are going through time-restricted feeding. It's a very beautiful, well-controlled study. And we are also collecting blood, so we can go back and check whether there are actually epigenetic changes or not. And that will give us some idea whether there is memory. Thank you.
Excellent. Thank you very much. Are there any other... Oh, I have some questions up there. David, you can just hold up. I like the enthusiasm.
Hi, thank you very much. David Kushinov, a PhD student at Center for Basic Metabolic Research. Apologies, my background isn't very much in metabolic science, but in medicinal chemistry. I had two quick questions. One is, you presented some data that in rodent models in an isocaloric environment in time-restricted feeding, you saw additional lean mass.
Yeah.
I was wondering, A, how this was measured, because I know that can be a little dicey, and any mechanistic explanations for the two? And then I had a follow-up.
So lean mass, because you're saying the small animal MRI may not be, because, actually we tried it in many different ways. We have also taken the muscle weight, and that improves. And then the functional state, the reason why we got into muscle was when we sent the manuscript initially, then one of the reviewers said, well, these mice are fasting for 16 hours, so can they actually run? Can they walk around? And then we put them on treadmill endurance test. And the result was super exciting and surprising. First thing, if you put high-fat fed mice on treadmill in the condition that we use, because it depends on the inclination and resistance, all that stuff, then the mice would run around 75 minutes. Sorry, the normal chow-fed mice. If you put the high-fat fed ad libitum, they run around 45 to 60 minutes in our assay. And if you put the time-restricted fed high-fat diet mice on treadmill, they ran for 120 minutes. We tried it again and again. So they always run for 120 to 140 minutes. If the time restrict for 8 hours, 9 hours, or 10 hours, when they go to 11 or 12, then they lose that endurance benefit. The second, in the grip strength, we also see time-restricted fed mice have improved grip strength. But anyways, grip strength doesn't have too much dynamic range, so we don't see that huge increase. So we are very curious about what is the mechanism of the muscle, and if you're looking for postdoc, that's your project.
If you need drug discovery scientists, let me know. And the other quick question is, I think we've seen lots of clinical trial data recently looking at the impact of time-restricted feeding in both isocaloric and non-isocaloric contexts. And you see, sometimes various improvements in metabolic health markers, maybe differences in weight gain and weight loss. Are you aware if there are metabolic ward studies in which you see these same kind of genetic or epigenetic changes? Where things are very rigorously controlled, isocaloric with just this time-restricted intervention?
Yeah, so there are some metabolic ward studies from Pennington, and where the time restriction was done only for a few days, and they didn't see much changes in RQ, 24 hours RQ, in those studies. But I guess what will be interesting is to do more flux analysis with stable isotope and a little bit longer time restricted feeding, and then see whether there is any impact. We actually have a grant just to do that on patients who have both inflammation and metabolic disease, because that's where we think that will closely mimic what we are seeing in mice, because most of the mouse studies are on high-fat diet, and these mice have high inflammation and also this metabolism. So that will be interesting to see. And going back to your comment about time-restricted feeding studies in humans, yes, there are many outcomes. The bottom line is, caloric restriction and quality of food. That field has dominated nutrition science, arguably, means for good reason, for the last 100 years. So then there are many established ways to monitor how many calories people are eating or what quality of food people are eating. When it comes to time restriction, since it's such a new field, most PIs think that you can just ask people, when did you eat your last meal? When did you eat your first meal? And that should be the gold standard to do this kind of studies. It's not. So that's why what you'll see is in studies where the participants were asked a question, did you do 8 hours time restricted eating? There you will not see an impact, because it's almost asking, it's almost, just imagine, if you randomize a group of people and then ask the other first group who are in caloric restriction, did you reduce your calories by 25%? If they say yes, will you take that for granted? No. So those are the kind of studies that are not showing any benefit of time-restricted feeding. And then when you combine caloric restriction and time restriction, particularly when you do extreme caloric restriction to 25%, then on top of that, seeing an impact of time restriction is very difficult in short term. There's a nice paper in mouse that came out from Joe Takahashi's lab in last week that showed beautifully that time restriction with caloric restriction can extend lifespan. But if you look at body composition, muscle mass, body weight, none of those factors actually show any difference between those groups. So that means there's some effect of time restriction that we cannot measure just from weight loss or some of the existing markers. So it opens up new ways to look at metabolic health. So thank you for that question.
Excellent. I think one last question. Quick question, quick answer.
Yeah, I'll be super quick. John Henderson, CBMR. I was interested, Zach said at the start, using monkeys as a potential model, because obviously the light-dark cycle and the nocturnal behavior of rodents is a big problem. So I was just curious, is that something you're going to try and recapitulate in monkeys, see if these different tissues have a circadian transcriptome under time-restricted feeding for a better model of a light-dark cycle?
So actually in 2018, I guess, we published a paper on baboon, multi-tissue transcriptome. So there were 64 different tissues in every 2 hours over 24 hours. And if you look carefully in many primate facility, actually monkeys are given 2 meals, typically 8 hours apart. So most of the primate facilities, most primates are actually on a time-restricted feeding schedule. And that might explain why it was very difficult to develop an obesity model in monkey, because they were always given high-fat diet within 8 hours. Only in certain primate facility, like California National Primate Research Center and then Texas, there the monkeys are, some monkeys who are living in the open, they are given some food ad libitum, but the food is very balanced. So coming back to your first question, actually in baboon, what we find is if we survey 64 different tissues, nearly 85% of protein coding genes show cyclicity, and it's very similar to what we found in time-restricted feeding in mouse. 2 very clear peak. One peak is around late afternoon, 11,500 genes reach their peak expression level. In the middle of the night, only 700 genes reach their peak expression level. And actually, what will be exciting is to see whether there are monkeys in captivity, because in US now, all the captive macaques are being sequenced. So if we can find, just like Karen presented, human genome, if we have 6,000 macaques that are in captivity in the US, we have their sequence. And if we find a mutation in reverb or any other gene, we can go back and ask, what is its impact on metabolism? Thank you.
Excellent. Thank you very much, Sanchit. And why don't we give Sachin and all this morning's speakers another round of applause. Thank you very much.
Thank you.
I have just a few quick housekeeping. So now lunch will be served outside the auditorium. Quick note, people who are going to be presenting posters should go and get their lunch first, because otherwise they have only about 25 minutes, maybe less now, before the poster presentation starts. So please, if you're presenting a poster, leave now. And then let's be back sharp for Ken Arnold's talk at 1:30. Thank you very much.